about is genetics versus epigenetics in crop breeding. You know, I work for the USDA, so we work with corn, barley, soybeans, that kind of thing. And you know, that's an applied science, it's applied genetics. So we'll talk about this subject in crop breeding, but also the broader subject, because crop breeding is really just artificial selection. But there's natural selection going on out there. So it's also an important subject in adaptation, plant adaptation, to say climate change and other things like drought. And there's the bigger, even bigger question of the role of epigenetics versus genetics. And we'll talk about the differences in evolution. And that's a tough subject. Now, I'm just going to work through the material. We're going to have fun. Actually, I have a section, hopefully. Actually, I have a section there in lectures that I give to 9 to 12-year-olds. And I actually think that it's really fun and it explains stuff, you know, in a way. If 9 to 12-year-olds are bright, and if they get it, certainly adults can get it, and they do get it. So the big question is how much in quantitative, I say quantitatively, in quantitative terms, not just is there a role for epigenetics or not, but how big is the role? Are there quantitative measures of how big the contribution of epigenetics versus genetics is to things like adaptation and evolution? So how much quantitatively does epigenetic versus genetic variation contribute to gain due to selection? Gain due to selection is if a plant breeder or a corn breeder is selecting for yield. How much progress, how much gain in yield do they see each year, year in, year out? That's gain due to selection. So how much does epigenetics contribute to gain versus genetics, to gain due to selection, or to short-term evolution? So if you think about it, if a plant breeder, a corn breeder, is selecting lines of corn that for yield, and that person is making progress, 5% gains in yield, that's essentially the same thing as short-term evolution. So if a population of plants is on the side of a hill, and global warming is occurring, and the average temperature is rising, how much does that population change, adapt over the course of several years? So often we think of evolution in terms of thousands, millions of years. But the fact is evolution is happening right now. It's happening in the short term. And I'll show you many cases of this, some cases of this. So short-term or rapid evolution versus long-term evolution, micro versus macro evolution. Whoops. Take-home lesson. There are examples in crop breeding. There are examples in crop breeding and in studies of short-term evolution that cannot be explained solely by genetic inheritance mechanisms. Basically, you know what genetic inheritance is: sequence changes. We've theoretically all have had genetics and learned something about this. You know, the basis of evolutionary theory is that there's a certain rate of sequence change in the genome of organisms, and that sequences which impart some favorable trait are selected for. That is the basic idea of you know, modern the modern synthesis evolutionary thinking, that the substrate for evolution are this slow rate of sequence changes that are occurring, and some change in the DNA sequence imparts some favorable trait, and that's selected for. The individual that gets that sequence change, that has that sequence change, is more reproductively successful. So that individual's offspring are more competitive, they spread in the population, they become more frequent, and that's how evolution occurs. But there are examples, a growing number of examples, and we'll talk about them, in crop breeding, but also in studies of plant and adaptation in the short term, in say 5 years, 3 years, that really cannot be explained very easily by this you know, modern synthesis genetic inheritance mechanism. And finally, in the introduction, I want to say that we cannot understand progress in discovery or acceptance of any of this stuff without understanding the sociology, you know, the psychology and politics of science. You know, literally how it's all about people. You know, the advancement in science is really a social interaction phenomenon. It's all about people, and whether an idea is accepted or not, or becomes broadly accepted, can be political and sociological in nature. And I mean, we don't have to talk about people when we talk about this stuff. We can just show the science. But I like that aspect of it. I enjoy talking about the people that are involved and what happens and how people interact. Certainly, over the course of my career, I've been insensitive to people's feelings. You know, I've been a little bit brash, certainly. I've had a big mouth. That doesn't help. If you want to be successful in science or in anything, you got to really fit in well. If you're socially like maladjusted, you know, it's going to hurt your career. So everybody you got to be really nice and very socially acceptable. Genetic versus epigenetics in crop breeding. So, lecture number 1. We're going to talk background today. We'll see how far we get, because as you could probably tell already, I tend to go on, you know. So, but let's just have fun. We'll talk about this stuff. We're going to talk about Darwin, Mendel, and Lamarck. The old modern synthesis, that's the modern synthesis, to 90% more, 95% of geneticists and scientists, I don't know what the number is, but to a very large number of scientists do not accept these new findings, have not absorbed yet what these new findings in epigenetics really might mean. So I call it the old modern synthesis, but it is still today the accepted way things work in terms of evolution. And the new modern syntheses, those that include epigenetics. So we'll talk about some basic mechanisms. We'll cover an example that I think is really fascinating about Linnaeus, Carl Linnaeus, the father of plant taxonomy, one of the most important biologists in the last 500 years. Very interesting, we'll talk about him, and one of his students found a mutant of a species, a plant species. And it was the first mutant ever described. In fact, they called it, the word for it is monster. It was, to them, a monster. And it turned out to be an epiallele of a gene, and we'll talk about what that is. So single gene, epigene inheritance versus multigenic inheritance. Important to understand what is multigenic inheritance and how might it play a role in what we're going to talk about. And then finally today, if I get that far, because I do babble a lot, I'm going to show you some slides from the Kids Tech. This is a weekend summer camp kind of thing, science camp, not summertime, but science camp for kids 9 to 12 years old that I've participated in recent years, and where I've talked about these ideas. And I just think it's fun and a way to explain some of the things we're talking about. So, crop breeding or short-term rapid microevolution versus long-term. Short-term, we're talking about 1 generation, 5 generations. So I'll give you an example of what we're talking about. Do you guys think that 1 generation, a single generation of exposure to some environmental change can alter the inheritance of the organisms that face that exposure heritably? What's the point of me giving these lectures? Why would you say yes?
Because of epigenetics.
Because of epigenetics. It's true. It's so interesting to me. It's fascinating. But anyway, 5 generations, 50 generations, 1 year, that's short term. It's so funny. I got to interject that when I've been giving this sort of lecture for a number of years, here for Daniel's class last 4 years, and just in the last 4 years, aspects of this that I'm going to talk about. A good example is something called paramutation, which we'll talk about, paramutation. 4 years ago, I presented ideas about paramutation, the role it might play in this phenomenon, and it was kind of like an out there idea, 4 years ago. So I left it to the end of the lecture, because this is like a weird thing that's, we're thinking about. But in just the last 4 years, now, if you read the literature, it's accepted science. It's a fact. It's no longer some weird thing. It's really going on, and people are, and there's more and more cases appearing about this phenomenon of paramutation. So things are moving rapidly in some ways. So short-term versus long-term, adaptation to changing environment. This is examples of short-term crop breeding or adaptation. Colonization of a new environment. This is a big field in plant biology. Crop breeding, but not speciation, not the creation of a new species. Over the course of time, the history of evolutionary thought was always about speciation, the creation of new species. I didn't have a chance, I didn't do this for this lecture, but I mean, that forces you to think about things in a certain way. The only stuff that's important in that model is stuff that causes new species. But what if that's not all of evolution? What if there's, changes, heritable changes within populations, that are significant, but not new species? Long term is hundreds of generations, millions, speciation and more, whole organism evolution. Think about it, evolution of, say, eyes or arms, big deal aspects of evolution. Mechanisms, short term, mostly epigenetic inheritance. And we'll give you, by the time we're done, we'll look at many examples. Long term, perhaps mostly genetic inheritance. So one way people look at epigenetic inheritance is that it's a soft inheritance, or inheritance is only important for a few generations, whereas genetic inheritance is more stable and could last over many generations. So, and finally, I don't know, we'll hopefully get to this. So we have this short-term phenomena, adaptation to climate change, response to selection of a plant breeder just in 1 or 2 or 3 generations, versus long-term phenomena. And a really interesting question is there a connection? And we'll talk about how this might happen. This is a totally open thing. It hasn't been shown yet. But is there a connection between epigenetic change, which we'll talk about, and long-term heritable genetic change? So, long-term macroevolution, Darwin, Mendel. Now, before I go any further, do you guys know everything about Darwin and Mendel and the modern synthesis? Or is that new to anybody here? Is that new to anybody here? You guys all know about Darwin and Mendel and the modern synthesis. Great. So there was Charles Darwin, and then along came, I visited where he worked. I'm a plant biologist, I'm a horticulturalist, I'm a pea person, a corn person. So for me, that was like a great experience to go there. There was a conference there, Barley Genetics conference there, right where he worked. Anyway, modern synthesis. So these two guys, and the combination of their views led to the modern synthesis, or neo-Darwinism, which was really the marriage of Darwin and Mendelian thought, and that with modern molecular genetics, with sequencing of genes. In the modern synthesis, the substrate for evolution are allelic, sequence-differing variants of genes. That's the substrate for evolution in this approach of modern synthesis. And evolution is via selection for or inheritance of favorable alleles or favorable combinations of alleles. And then there was this guy, Lamarck, and Jean-Baptiste Lamarck. And he was before their time, somewhat before their time, but not much. But his ideas, and we've all heard about this, it's the giraffe reaching for the higher and higher branch. And the complexity, he had 2 big ideas: the complexifying force, or the force that perpetually tends to make order, sort of like anti-entropy. And his second idea, so the complexifying force, everything is getting more and more complex. And the second idea, this is the one that's relevant here mostly, is the inheritance of acquired characteristics. Okay? In this model, organisms sense their environment and adapt to it in a heritable fashion. Adaptive phenotype, perhaps determined by inheritance of epialleles, which we'll talk about. Same sequence, but different programming. So I'll give you a definition of epigenetics. Epigenetics is, well, it's actually a difficult term, because if you read the literature, it's a term that lots of people think is misused or overused. It stands for a lot of things. But just for our purposes, we'll use the definition that epigenetic inheritance is inheritance of differences in the regulation of genes, not in the sequence of the genes themselves. And we'll talk about ways this can occur. So the sequence of the gene is not changing, but the regulation or expression of that gene or many genes can change. And that change in regulation is heritable. And I'll tell you right now, it's happening. There's a lot of examples out there. There's a growing literature. It's happening with plants. It's happening with animals. We'll talk about many examples of these things. Yet it's still very controversial. It's very controversial. We've tried to raise some funding. Daniel's worked with me so that I could get some projects going here in Portland, since I'm relocated here. But if you read the reviewers on our grants, sometimes they're really hot and bothered about some of this stuff. So it can be still very controversial. That controversy might die down over time, who knows, as it becomes more accepted. That's the way things work. At first, new ideas are very controversial. Some time goes by. It's clear that they really are happening, and there's more acceptance. It's a sociological process. And I can remember in my career, when I in a completely different field, I made some progress. This is many years ago, 30 years ago. And for the first 10 years after I had made this advance, it's just, there was no traction for it. But after about 5-10 years went by, oh, okay, this is interesting, this is good, we'll support it, we'll fund you. By that time, 10 years had gone by, I was no longer interested in that work, that particularly. Anyway, so Mendel's laws and Darwin's ideas led to a happy marriage, modern synthesis. Darwin actually was very familiar with Lamarck's ideas, the inheritance of acquired traits, and he appreciated it. Darwin did not reject Lamarck. He appreciated it, but Lamarckian ideas were really rejected and led to some pretty bad stuff. I don't know, I can't do too much history of science here, but there was something called Lysenko, a person called Lysenko in the Soviet Union, who was the accepted form of genetics in the Soviet Union back in the 20s, 30s, and so on. Scientists, geneticists, got sent to the gulag. I mean, people actually died over this stuff. So there were really some bad outcomes, and once you have a negative outcome, it could stick with you for a while. So if an idea is discredited, it's hard to get away from that. It takes time for things to be accepted. So I have a but here, because recent, as I'm going to show you, significant revival of interest in this guy's ideas. I had one of the first year I gave this lecture a few years ago, a student came up to me after, or emailed me, saying, are you saying that Lamarckian inheritance is happening? And I said, yes. And it was difficult. Recent significant interest. Are some modes of epigenetic inheritance really Lamarckian? The answer is yes, they are. So if new knowledge of epigenetic inheritance is truly transformative, the funding agencies, I like to make these lectures fun, but if any of you go on to a career in science, which I don't recommend, it's very difficult. But if you do go on to a career in science, you have to get funding, competitive funding. And the agencies like NSF are very interested in funding work that is transformative. That's one of their favorite words, transformative work. The problem is they recognize that they has a bias in the reviewing process against truly transformative work. This is recognized. I'm not making this up. They actually did a survey about this a few years back. So they want to fund the transformative work, but at the same, see, I'm rambling now. Ranting is the right word. They want to fund transformative work, but they're biased against transformative work, truly transformative work. But anyway, if this new knowledge is transformative, meaning it will have a great impact on our understanding of stuff, inheritance, adaptation, evolution, then this discovery, this transformative new knowledge will lead to new discoveries of mechanisms of inheritance that we don't currently understand. If the new science is transformative, it's going to lead to new discoveries which are not anticipated, that we did not anticipate. An example of that, actually, I think a fair example, a good example, is this phenomenon of paramutation, which I'll go back to. This phenomenon actually happened in a gene that I didn't discover the gene, but I was the first to define that gene. And it actually is happening at that gene, and we're studying it. Very interesting stuff. So if you have a really new thing, it's going to lead to really new stuff that people don't initially understand, and take some time to figure it all out. So I hate to go on and on, but let me just mention Goethe, who was the individual, one of the first humans on Earth to get evolution, to get the idea of evolution. And he said, and he loved plants. He studied plants, and the ever-changing display of plant forms, which I have followed for so many years, awakens increasingly within me the notion the plant forms which surround us were not all created at some given point in time and then locked into the given form. This is in 1831. They have been given a felicitous mobility and plasticity that allows them to grow and adapt themselves to many different conditions in many different places. And that is exactly what we're going to be talking about. How does that actually work? How do populations of plants have this felicitous plasticity, as he called it, because they do, you know. And can this change over time be explained solely by sequence differences, not other kinds of differences? So this was one of the papers that we put online. You could look at it, but I want to just quickly review it and to show you that these things that I'm going to talk about are controversial. There's no consensus today. So there was this commentary in the journal Nature. One reason why I like to give this as an example is Nature is the #1 journal of science in the world, you know. And so these are not shabby folks. These are all established scientists. And does evolutionary theory need a rethink? Researchers are divided over what processes should be considered fundamental. One side of this debate, this commentary, it's a debate, is yes, we need to urgently rethink how we view adaptation and evolution. The above insights derive from different fields, but it fits together with surprising coherence. They show that variation is not random. Whoa, just think about those words. Variation is not random. That's the whole modern synthesis is based on, the slow background rate of random variation. Variation is not random. What does it mean not random? I mean there's a process that causes variation non-randomly? That's Lamarck. And that's what people don't like. And it is difficult. That there is more to inheritance than genes, and that there are multiple routes to the fit between organisms and environments. Then there's the other side. No, all is well. What Lalonde and colleagues deride, and what now deride is a strong word. Deride, put down. To put down. Deride as a gene-centric view. Oh my God, I've been guilty of using that very term. And I faced up to that term in my own work. Because when there's a strong paradigm that explains things very well, which is what they're talking about, then it's hard to shift that paradigm. The stronger the paradigm, the harder it is to shift it, to move it. And the gene-centric paradigm, Darwin, Mendel, DNA sequence, the genome, hugely strong paradigm that explains an awful lot. Hard to shift. So this gene share would de-emphasize the most powerful, predictive, broadly applicable, and empirically validated component of evolutionary theory. Sorry for talking so fast. There is little evidence for the role of inherited epigenetic, listen to this, little evidence for the role of inherited epigenetic modification in adaptation. We know of no case in which a new trait has been shown to have a strictly epigenetic basis divorced from gene sequence. They're saying, and this was 2014, I don't have the date up here. They're saying that there is no case for an inheritance of a phenotype, of a trait, that is not, gene sequence differing have a basis in gene sequence difference. Well, they say here a new trait, and I was thinking about it recently because I use this slide. So I say maybe epigenetics doesn't lead to an entirely new trait. So maybe epigenetics doesn't lead to a different floral structure or something like this, an entirely new trait. 3 arms versus 2 arms, But there are definitely epialleles that do modify, alter, adjust already existing traits. And we're going to talk about them. But come on in. It's okay.
Oh, no.
Okay. You don't have to come in. No. Back to this. There is no, where did they say that? Somewhere in here they say we know of no case of epigenetic inheritance. Uh-oh. Here's a case. A single generation of domestication heritably alters the expression of hundreds of genes. That's why I asked you about the single generation. I'm surprised so many of you think that it's possible. But you have to understand the mechanisms, A single generation of domestication heritably alters. What this study did is they have trout. This is local. This is a trout farm in the, not far from here. What they found is that when they take, baby trout in a hatchery and grow it in a hatchery, a fish hatchery. So it's basically growing it in a fish pond, for those who don't know about it. They found that the fact that these fish were growing in a different environment than the wild, that there was lots of changes in gene expression. Changes in gene expression that had to do with adaptation to this different environment. Because think about it, if you're in a little fish pond versus the ocean, you're much closer to all the other fish. There's crowding, injuries that occur, food differences, all kinds of differences. And they found that there were 723 genes that were differentially expressed due to this growing up in the fish hatchery, say a human growing out on a farm versus a human growing in a city, or a human growing with very little food versus a human growing with lots of food, an environmental change. And they showed that they were transmitted to the offspring. In the very first generation of these animals are undergoing gene expression changes that are adaptive to their environment. Those changes are transmitted to the offspring. So here's an example of rapid evolution that probably involves a single gene trait. I'm going to talk a lot about single gene traits that are determined by variation of single gene versus multigenic traits, and how it applies to this area of work. Rapid evolution of flowering time by an annual plant in response to climate fluctuation. So what they showed, that there was evolution that they think is, we're going to get into a confusing issue. One of the problems, I think, with people understanding or accepting new ideas is the use of words, the sloppy use of words. So when you're talking about evolution of flowering time, well, flowering time changes didn't evolve. There was slight changes in flowering time. I mean, is that evolution of flowering time? Yes, it's change of flowering time. But you see that it can get problematic if you're not careful with your choice of words in describing these phenomena. Because somebody will say, well, wait a second, that's not evolution. That's just this population is adapting over a few years to a difference in flowering time. It's not evolution. Who knows? Maybe it is. But anyway, these authors report evolution of flowering time to avoid drought that occurred from 1987 to 2004 in a population of Brassica rapa. Brassica rapa is like a cabbage plant. No, is that right? Brassica, yeah, like a cabbage, a species of cabbage. Anyway, and so they had this population, and there's global warming, climate change, things are getting drier and drier, and they found in this population that the flowering time was shifting, so the plants reproduced a little bit sooner to take advantage of moisture that was there. And this was a heritable change. But it occurred only in a few years, not 10,000 years or 5,000. It's happening in the present. How could that work? Well, it turns out that flowering time can be determined by one or a few genes. So all you really need in this case is a slight change in expression of one or a few genes, and the flowering time can be changed. So that could be a relatively simple trait. Is it natural selection? It is natural, right? This is not a lab or greenhouse experiment. This is something that's happening out in the population. Is the altered flowering time due to modulation or change of one or a few genes? It is I say single gene trait, but it can be one or a few genes, not many genes. Do plants in this population sense the climate change and have a mechanism to heritably alter their programming and transmit the new programming to their offspring? Is this heritable change induced in response to the climate change? Think about what I'm saying here. Or is it random variation? This is the core of the issue here. So that's an example. Two examples, one with fish, one with plants, of rapid change, heritable change in organisms that is occurring. Environmental epigenetics and the unified theory. So this was a sort of an idea paper about this stuff. But look at the title, a neo-Lamarckian concept that facilitates neo-Darwinian evolution. So, while it might be controversial, this is a hot subject area in biology. So, how do we incorporate this new science that is bubbling up into models of how adaptation and evolution work? And this is a figure from his paper where he's talking about marrying Darwinian theory with neo-Lamarckian concepts. to create a new synthesis. Now, the modern synthesis, neo-Darwinism, was referred to as the modern synthesis. So you talk about tricky words, use of words. Modern. So this is a new. That's why I had this old modern synthesis and new modern. So this new modern synthesis incorporates this kind of epigenetic change that people think of as how organisms or populations respond to environmental change. What do most people think is the most important development in genetics? Important to say biomed. If I asked you guys, forget this epigenetics, blank your minds out. What's the most important thing that's happened in the last 10, 20 years in plant biology, et cetera? The answer is genomics, genetics, DNA sequencing, sequencing the human genome. Remember how much, well, maybe you guys wouldn't remember, it depends on how old you are, how big a splash it was that they obtained the human genome. Okay, the idea was when those companies finished sequencing the human genome, this was like, oh my God, we know it all now. That's the feeling that was communicated. We can explain everything now. We have the genetic code of humans. But it turned out that it didn't explain everything. It turned out to be essential to have it to eventually explain everything. So you need the genetic code, but then you need to know how it works, how it's programmed, how its programming is inherited. That's the basic idea. So we now have the entire genetic code of model species like Arabidopsis, major crop species like rice, corn. We've gone beyond that. So we have the genome sequence of a bunch of important species, many species, plants, animals, and we've gone beyond that. The power of this technology keeps growing. So now there are cases where there are studies where they sequence the genomes to get the genomic sequence, not just of a species, but of many lines, and figure out how their genomes are changing over time. So lines, varieties, land races of a given crop have been sequenced. This new knowledge and technical power is already generating advances, many advances. So genomics, genetics, classical genetics is important. It does provide advances in science, practical advances, and just new knowledge. But the potential of genomics might be limited. Why? Because of the contribution to phenotypes of so-called hidden heritability. This is what I'm talking about, epigenetics. Possibly hidden because of the contribution of the epigenetic code. So this was a review that came out, Hidden Inheritance in the Human Genome Project. Researchers couldn't determine the genetic basis of many diseases. So the idea was that they spent a lot of money sequencing the human genome. And now that they spent all this money, let's explain the genetic basis of all these heritable diseases. But it turned out that they couldn't explain enough. They couldn't explain enough to satisfy the people who funded them. So what's going on? Somehow the human genomicists were missing something. What they were missing was epigenetics. Heritable change in phenotype and function caused by mechanisms other than change in underlying DNA sequence. Now, there's not just one component to epigenetic inheritance. There's several. If I have a chance, I'll tell you what's popular and what's not in this field. Because some popular things actually might delay progress. So here's a popular thing, heritable and reversible DNA methylation. And we'll talk about that. That is probably the #1 thing people are looking at in the field of epigenetic inheritance, is changes in DNA methylation. We'll talk about what that is and mechanisms, et cetera. But you know what? That introduces some problems. And we'll maybe get to that. Changes in histone modification. Those are the proteins that bind to DNA, to genes. And depending on how they're binding to the genes, to the promoters of genes, they could either function to turn on or off a gene. And you could get actually transmission. I didn't think of showing you that paper. You could actually get heritable transmission of histone modifications, which is very interesting. And then, of course, gene and gene network regulation via complex interaction of numerous proximal and distal regulatory elements. Enhancers, silencers, insulators. Finally, if we get a chance, if we get that far and I don't babble too much, we'll talk about RNA-mediated transmission of epigenetic programming, which is a very important thing. But let's just for a moment think about #3, gene and gene network regulation. I can explain it to you in about one moment. If you could picture many chromosomes in a nucleus, in the human 23 and corn 10, and it turns out that the genic content, the genes, represent a small fraction of the total genome. You've probably heard about this. 1/10 or less of the sequence of all the chromosomes are actual genes. All that other stuff, transposable elements, intervening DNA, all that other stuff, is full of little short sequences that are like the on-off switch for genes. And I'm going to give you an example of this later on. You could have genes might be 5 kilobases in length, say, 5,000 bases in length, a gene. These guys might be 20 bases in length, or 50 bases, or 10 bases. And they might be right next to the gene, or they could be very far away from the gene. They could be 100 kilobases, 100,000 bases away from a gene, but yet they are the on-off switch that turns that gene on or off. And you have interaction of all these elements with each other, a massively complex programming mechanism. And that's really what's going on. That's why we are who we are. It's very complex. Lots of room for study. So you guys know all about this stuff, the double helix, the 4 bases of the genetic code, and we'll talk about how cytosines can be converted, one of the 4 bases, to 5-methylcytosine in the central dogma of DNA, of the molecular biology. This describes the flow of genetic info. You have a gene, it's transcribed into a messenger RNA, which is then translated into a protein. And your typical gene looks something like this. It's got sequence which encodes a protein, but then it's got a promoter, the on-off switch of the gene. And this is where this promoter, which can be right next to the gene, part of the gene, or far away from the gene, but it's the regulation of this gene's expression through this promoter, this on-off switch or volume switch, which is what's going on here. And the thing is, if a trait is determined by one gene, easy to think about turning up the volume of that gene's expression, turning down the volume of that one gene's expression. But what if a trait's determined by variation in expression of 100 genes? It's more complex. And the fact is, that's what's going on. So yes, there are single gene traits, but mostly what we're talking about in terms of what a phenotype, what an organism looks like, and how it responds to the environmental change, is subtle change in expression of many genes together through regulation. I'll give you a simple example. There's something called transcription factors. And you can have a transcription factor which regulates a master gene or a few genes, which then downstream regulates hundreds of genes. So, the programming can be very complex. So anyway, you guys know all this stuff. So a gene is a short piece of DNA and 5,000 base pairs or so in length, but the genome is huge compared to that. The common bean genome is 650 million bases, or enough for 130,000 genes if they were all 5,000 kb. The corn genome and the human genome are about the same size, 3,500 megabases, or enough for 700,000 genes. A Arabidopsis genome, which is, I don't know how many of you guys are familiar with who knows what Arabidopsis is here? Some of you. Well, it's a model species for plant research. Has a small genome size, and people do lots. It's easy to grow. You can do a whole experiment on a bench this size in a greenhouse. Handy to grow. So these chromosomes have genes separated by a lot of other stuff, or gene islands, then interrupted by other stuff. And that other stuff turns out to be important. It used to be called junk DNA. It's not junk. For a while, people thought this non-genic part of the genome was junk DNA, but it contains transposable elements. DNA that is non-coding, that is transcribed into non-coding RNA. Gazillions of very short sequences that are enhancers, I mentioned to you. And all this other stuff functions to regulate in a very complex manner. Not just one or a few genes, but many genes. So if there is an artificial or natural selection, does it work via heritable changes in one or more genes, or in this other stuff, or both? Does evolution, does phenotypic change, heritable change in phenotype due to gene regulation, work through the regulation of one gene, many genes, or all this other stuff throughout the genome? Well, the answer is both. It's a very complex story. And to really get a handle on it, you have to be working in biomed. That's where you get, if you want to go into science, go into biomed, because that's where the big bucks are. Although some people have gotten big grants lately that could pay for a lot of sequencing. But boy, it could get expensive. So, DNA methylation. Cytosines get converted to, see that methyl group right there? You could pluck on a methyl group to form 5-methylcytosine, the so-called 5th base of the epigenetic code. Now this change from cytosine to 5-methylcytosine is reversible. And one of the difficulties of epigenetic inheritance is that it's hard to study. A sequence change is stable. If you have a mutation, and many scientists have done mutation genetics, where you look for mutations in genes that you're interested in, and you get a sequence change, and it knocks out the gene, and then you could study the phenotype, etc. Mutation genetics. Whereas an epigenetic change could be clearly heritable or unstable. This could happen in a plant, and then you grow out the plant, you self-pollinate the plant, and get the seed for the next generation. Whoops, that change is now gone. It's changed back. It's inherently unstable, which does make it hard to study. Really, the same is true of histone modification, which is the second component of this code of epigenetic regulation, that you get modification of histones, which either turn on or turn off genes. And let's look at one example to start with of DNA methylation. So we now know that there are epigenetic alleles or epialleles of single genes. Now these are either highly methylated, so that's hypermethylation, or hypomethylated, so where the methylation patterns are erased. The most common version of this is having a gene that is active is hypomethylated in certain sequences, and then to turn that gene's expression off, you methylate it. So the most common thing is that methylation turns a gene's expression off or on. And the first example I'm going to give is this Peloria mutant discovered by a student of Carl Linnaeus. So this Peloria mutant is inherited as a single gene variant that alters the flower structure of the common toad flax, or Linaria vulgaris. It turns out that this mutation was in 1744 and is today the result of inheritance of an epiallele. Now, got my fancy new watch. We got a few more minutes. We can keep going. What time are we finishing?
30.
Okay. I'm not sure I'll get through it all, but it's okay. I talk too much. I'll skip over the background about Carl Linnaeus. I want to get to my 9 to 12-year-old lecture slides. I'm going to skip over this. Bottom line is the point about Carl, you can look at it online, is that he was the father of plant taxonomy, and he believed that all plant species were formed in an instant by God. Yet his student discovered an exception to that. And the other interesting thing about it is that he determined the taxonomic relationship between plants by floral structure. How many petals, plant had.
Isn't he why we named genus, species?
Yeah, it's him.
Yeah.
That's him. That's him. That's Carl Linnaeus. Although no scientist works alone. So there were others going on. This was developing in the community of science. But basically, yes, that was him. Genus and species, Homo sapiens. But look, they discovered that the typical structure of this flower has a dorsiventral, asymmetric, bilaterally symmetric flower, but that there were some lines which had a very different structure. And they called those lines floral structure, a radially symmetrical floral structure. And they called that mutant, who was the first mutant ever described, the monster. That's what Peloria means. It was a monster to them. It contradicted their worldview, his worldview. A species is a species. It's not changing. It was created by God in an instant. And we could separate them and define all their relationships by their floral structures, which are fixed in time forever. Uh-oh. But now we got the appearance of this guy, this mutant, this monster. How could that possibly be? Turned out that the wild type is hypomethylated throughout this gene which controls this floral structure, and the mutant is hypermethylated, shut off. So that gene shut off in the mutant, and the fancy bilateral, so when the gene is expressed, the flower has the bilaterally symmetrical flower. When the gene is shut off, it's radially symmetrical, and it's just due to methylation of the gene, flipping it on and off. And that was proven just in 1999, not too long ago. Now, I could see that as much as I talk and as long as I go on, I'll never get through all the material. But let me show you a couple of things before we quit today. And if it's 12:35, I'm going to give myself to 12:25 and then move right on to the talks, the slides for the 9 to 12-year-olds, which I think are very fun. Typical Mendelian inheritance. This plant is purple. Purple corn plant is homozygous for the big B, the B gene. The homozygous recessive plant is green. It's homozygous for the recessive allele, little b. And this is a gene in the anthocyanin pathway in plants which determines plant pigmentation. If it's homozygous big B, it's purple. Homozygous little b, recessive. The heterozygote is big B, little b. You guys should have had this somewhere. How many people, Punnett Square, all of you. Big B, little b, and you self that guy, and you get 1 to 2 to 1. 1 big B, 1 big B, little b, and 2 big B, little b's, and 1 little b. That's Mendel's first law, segregation of alleles. Mendel's first law. And we as a society love our laws. Whoops. Okay. Wait, what did I do? Oh, I was just going to show you that for a reason. And the final piece to this puzzle. We're going to come back to that because I think I want to show you this. I'm going to skip over that. I'll get back to the quantitative genetics next time because I want to show you something. No, I have to go back. It is background, essential background. So, but for some genes, here's the final piece of the puzzle. Single gene traits versus multigenic traits. For multigenic traits, quantitative traits, at first, people could understand single gene traits, but in terms of evolution, it didn't make sense. Because most traits, in terms of evolutionary thought, were continuous traits. Traits, that varied from short to tall. And, but this guy here, R.A. Fisher, showed that if you had single gene inheritance, but for many different genes that affect the trait, that single gene inheritance for many genes, across many genes, Mendelian inheritance, but for many genes, can result in a continuous phenotype that could explain multigenic inheritance. And now, Mendel and Darwin could live happily together. Because Mendelian inheritance, which is round versus wrinkled, green versus yellow, now that single gene inheritance, if looked at across multiple genes, can explain continuous traits. And the evolutionists like that. Now, Mendel married with Darwin, explains it all. And skip that. I want to skip all this, how he got to this idea. If you see single gene, 1 to 2 to 1, and then you have 2 genes affecting a trait, you add up all the alleles. Then if you keep adding more genes that might impact the trait, eventually you get to a continuous phenotype. And that's, now everybody's happy. We could explain inheritance and evolution. Now here's an idea. I've shown you an example that a single gene trait can be epiallelic. The flowering trait that changed the structure of the flower was an epiallele of a single gene. But think, if single gene allelic sequence variants can add up to multigenic traits, and if single gene epiallelic variants are heritable and can contribute to a phenotype, then single gene epiallelic variants might add up to multi-epigenetic or epigenomic traits. If you could show that single genes affect traits, but then if you take the single gene variation at many genes, you could get quantitative traits. Then you show that epiallelic variants of single genes can determine traits. Then perhaps the combination of epialleles at many genes can determine traits. And I think, in fact, this is what's happening. I'm going to skip that. So all this progress led to, oh, here we go. I did get there, you see? Look. And the next bunch of slides are fun slides. So I could slow down and explain this better. So, because I am getting there. The progress of science. First, there was Mendel, but he was not initially understood. He was rediscovered, as you probably all heard, by a guy named Hugo de Vries. There was others, around 1900. Then scientists showed there was the chromosome theory of inheritance. Then Thomas Hunt Morgan proved that genes are on chromosomes, like beads on a strand. Then Oswald Avery showed that DNA was the material of inheritance. The chromosome was a strand of DNA. Then along came Watson and Crick. And you've all heard of Watson and Crick. Now I showed the real heroes. These Watson and Crick, well, Crick was brilliant. Watson. He once axed my application for a job, so I will never like him. He did. We can't go into that now because we want to have fun. But these guys, Linus Pauling, Erwin Chargaff, Rosalind Franklin, she's one of my great heroes. They were all critical to this discovery of the structure of DNA. Rosalind Franklin, very critical. Oh, Linus Pauling, totally critical to this. But was he on the Nobel Prize? Well, she had passed away, so she could not, because you're dead, you can't get the Nobel Prize. She was really something. I'd love to go on about her, but I won't. It's a great example of a women in science and the issues that affect women in science. So she was a woman in London, in King's College, in a lab dominated by men, in a world dominated by men. Which is, I was going to say, okay, well, whatever. All this progress of those guys led to the modern synthesis. But here's my view. This is the point about paradigms and how hard it is to introduce new knowledge, new science. Because first there was the overwhelmingly powerful paradigm of the gene, Then, the gene-centric view. Then there was the genetic code, the genome. Wow, now we have the whole sequence. But perhaps it's not the gene complement of a genome per se. Perhaps it's not our genes per se. Now, again, genes are important. Genetic sequence differences are important, obviously. But perhaps it's not the genome per se that's important. Rather, a genome's collection of genes are like a piano, the keys of a piano. And it's not the piano itself, but how it's played that's important. That gets me to my fun slides. Always like to have fun. Give these lectures for these kids. I love it. Okay, so I explain to them that the genetic code, is it the piano that's important or the music played on it? And then I tell them that I could explain how all life works in 30 minutes. I think I can. Okay, so Mr. Spock, he says the genome of this individual. He gets it using this analytical device. It's so funny because that's exactly what's happening. And then he uses that same analytical device on these two and he says, Jim, they have pretty much exactly the same genetic code. Remember, I'm talking to 9-year-olds here. They have the same genetic code. How interesting. They look so different, yet they have the identically same genetic code. This is one of the reasons that I, as a scientist, am so bothered by racism in our society, and it's so ignorant, because we really are almost identical. We humans are so well adapted to seeing differences between each other. We're real good at that. We've evolved that ability to see differences. That's important for survival, right? But it prevents us from seeing the similarities. So, Officer Spock, hey, Jim, the genetic codes are 98% the same. How could they be so different? Oh my God, all these different organisms have largely the same genetic code. Now people say to me, how could a plant and a human have the same genetic large? The question is similarity versus difference. Are they more similar than they are different? The answer is yes, they are more similar than they are different. Differences are important. Sure, it's important that it's a plant or a cow or a fly. But are they similar? I'll give you a brief example, because we all got to go. A pathway that I've worked on in my career, it's a biochemical pathway called the inositol phosphate pathway. Turns out that that pathway is essentially identical in all these different organisms. So if you were just studying that pathway, you'd say, these organisms are really very similar, not very different, right? So anyway, so this is the fun part. How many genes does a piano have? How many keys does a piano have? 88. Yet one piano can make so many different types of music. Oops. Oh, I screwed it up already. Oh. How do I? Wait, I'll get this. Look. Oh, I don't have it. Oh, you can hear it. It's okay. They can hear it, right? No, let's not bother. Classical music, rock and roll. This is for kids, but remember, I can't listen to that and talk to you at the same time. So I guess, to be fair, the quality of the piano can be important. Genes are important. But really, to me, it's not the piano, the genome that's important. Rather, it's the piano player. And that's what we're going to talk about. We're going to look at specific cases, let you guys go. But here's a brief example. If you want to look at these papers before Wednesday, turns out that if an individual, for example, a person, a human being, is obese as a child versus not obese, that different. In other words, if an individual grew up in a time when there's plenty of food, actually it's not obese. I said that wrong. If an individual grows up at a time when there's plenty of food versus another time when there's a famine, that the grandchildren will have a decreased incidence of cardiovascular disease and diabetes if their grandparent was lean versus fat. Think about that. Now, that's important to me. In a society where obesity is a real issue, what's the big health question? Well, certainly to address obesity in our society. What about the fact that, based on science, we could say that 2 generations later, there's going to be an increase in cardiovascular disease and diabetes, a big increase in cardiovascular disease. Now, if I was a pharmaceutical company, maybe I'd want to know that and plan for it. But they don't think that far ahead anyway. They think 1/4 of a year, 4 months, that's it, 3 months. But, so this is important. What if you're a plant breeder and you're interested in breeding plants for drought tolerance? That's actually a very important subject, right? We're undergoing global warming. What if you're using methods that follow the inheritance of genes, of genic variants, but there's a whole other phenomenon going on, adaptation to drought in all the plants, that you're ignoring? Might that impact your breeding success? We'll talk about this next time and the time after. Thanks for your patience.